Hello Year 10 and welcome to the first ever quickfire question quiz. Now if you haven't read the instructions already, here's what we're going to do. We're going to find a group of people to Zoom chat, house party chat, FaceTime, group WhatsApp call, and we are going to have ourselves a quiz. We've got 50 questions. We're going to do it like we do in the classroom, as if we've got our mini whiteboards. I'm going to say the question, you're going to shout the answers to each other, and you're going to score your own points. So if you haven't got your group together, you haven't got your Zoom party ready, then sort that out now. Okay, so here we are, we're on B1 Quickfire Question Quiz, and we are going to look at some questions commonly asked, short answers for this topic, okay? We're going to do this every week for topics 1 to 4. And you can see that I've got my Quizmaster voice ready. We are ready to go. Okay, first question. What happens in the ribosomes? Question 1. What happens in the ribosomes? Now, you might choose to put your whiteboard up to show each other. You might be shouting the answers out. And hopefully what you've got is protein synthesis. And synthesis is a fancy word for saying making. So the making of proteins happens in the ribosomes. Okay, hopefully you're getting a flavour for where this is going. We've got 50 questions. We're going to work our way through. If you haven't done some revision, you might want to revise this and then come back to this. Who knows? Hopefully you're not that one person who's watched the video before the Zoom chat, remembered all the answers, and then played with their friends, because you will get found out. Okay, question 2. How do bacteria reproduce? Do they use mitosis? Do they use the cell cycle? Do they use meiosis? Or is it something else? How do bacteria reproduce? Question 2. And the answer is binary fission. Okay, it's by binary fission. Question 3. What is moved through the xylem? What is moved through the xylem? So we always remember xylem and phloem together. And what is moved through the xylem? Possible 2 points available here. Okay, let's look at our answer. Water and minerals. I'll give you 1 point for each. Okay, so water and minerals. It's the process of transpiration, moving in a singular direction. You might say one direction, from roots to leaves. Transpiration. Question 4. What is the function, and function means job, of the chloroplast? So what does the chloroplast do? Okay, what is the function, the job of the chloroplast? Question 4. And the correct answer is the site of photosynthesis, where photosynthesis occurs. Okay, that idea of the plants making their own food, that glucose, through the process of photosynthesis takes place in the chloroplasts, and they're floating around in the cytoplasm. Question 5 on the quickfire question quiz. What do we mean by cell differentiation? What do we mean by cell differentiation? I've got the word different at the beginning there. What do we mean by cell differentiation? Okay, shout your answers at each other. If you're playing on your own, then just shout your answers at the screen. Okay, here we go. The answer is when cells become specialised. They're becoming different. They're specialising into those specialised cells. Question 6. What is the first stage of the cell cycle? What is the first stage of the cell cycle? So there are 3 stages. Name the first one. Oh, this is a tricky one. Do I need to give a clue? I'm going to give a clue. It begins with an I. Okay, begins with an I. And the answer is interphase. Spellings don't matter if you're shouting out. Spellings definitely don't matter. Okay, question 7. What is a stem cell? What is a stem cell? Hopefully you've done some revision before we've come to our quickfire question quiz, but you might be thinking to yourself, oh yeah, I've heard of a stem cell. And that means you probably didn't do the revision beforehand. Okay, so what is a stem cell? And it is an undifferentiated cell, a cell that has not yet specialised. Okay, so you can have 1 mark for using either undifferentiated or unspecialised cell. Question 8. Which 3 parts of a cell are in a plant cell but not in an animal cell? So an animal cell is really quite simple. We've got to name 3 parts which are in the plant cell but not in the animal cell. Okay, and let's look at our answer. We've got the cell wall, keeps the plant cell rigid, keeps its shape, supports it. We've got the chloroplast, we've met that already, that's where the photosynthesis takes place. And we've also got the vacuole, contains the sap right in the centre of the cell there, that sticky sap that we would find in plants but not in animals. 1 point for each correct answer. Moving on, let's go to question 9. What is the movement of particles from a high concentration to a low concentration called? Movement of particles from a high concentration to a low concentration. Hopefully we're all shouting one of 3 words, and hopefully we've gone for the correct word, which is diffusion. Okay, moving on to question 10, let's have a look. What happens in the mitochondria? Mitochondria is that long word beginning with M. And what takes place in the mitochondria? They're floating around in the cytoplasm. Shout out your answers. And of course it is respiration. We're a 5th of the way through our quickfire question quiz. If you think that this is time to have a snack, maybe give it a pause and have some snacks, have a chat, and then come back to me. Let's move on to question 11. Maybe my quiz voice is a little too quiz-like. What are the 3 sources of stem cells? Should be an S on the end, sorry. What are the 3 sources of stem cells? Actually, maybe it doesn't need an S. I think we're okay. Okay, shout out your answers. Write them on your whiteboards. Shout at the screen if you've got nobody else. That's okay. Okay, 3 sources of stem cell. 1 mark, bone marrow, also known as adult stem cells or adult bone marrow. We've got embryos left over from IVF waste, and we've also got umbilical cords. That's our new technology. Can't be used for us, our umbilical cords are long gone, but could be used for your future children. Okay, 3 sources of stem cell, 1 point for each. Moving on to question 12. What does active transport need? Now this can be interpreted in a couple of different ways, so potentially 2 marks available here. What does active transport need? Question 12 on our quickfire question quiz. Okay, and the answer is energy. And also the energy is used by those carrier molecules. Okay, they pick up the particles and they move them across. Okay, moving on to question 13. What is the second stage of the cell cycle? What is the second stage of the cell cycle? Okay, the second stage of the cell cycle is that big stage. It is indeed mitosis, one that we have to spell correctly in the exam. Okay, moving on to question 14. What is the movement of water from a high concentration of water to a low concentration of water called? So we're moving the water. It's going from where there's lots of water to where there's not as much water. What do we call that one? Movement of molecules. And for 1 mark, we've got osmosis as the answer. And moving on to question 15. How are the alveoli adapted for diffusion? We have 4 possible answers here, so we've got 4 possible points. I'm going to give you a little bit of time. Think those points through. Just realise that that carrier molecule could also be called a carrier protein. So if you did write carrier protein earlier, that's also fine. How are the alveoli adapted for diffusion? Okay, we're going through those answers now. There's 4 possible points here, 4 possible score points. Large surface area, it's our classic friend in any biology situation. Large network of blood capillaries, lots of places for all of those substances to go into the blood. We've got the moisture there so that all of those gases can dissolve before they move across. They must be soluble and thin, 1 cell thick, in order to get those substances across. So that's question 15. Moving on to question 16. Don't forget, 1 point for each there. How does glucose move into the blood at first? So we're talking about glucose moving from the small intestine into the blood. How does the initial movement, first of all, of glucose go from the small intestine through those villi into the blood? Okay, and the answer is diffusion, because there's a high concentration in the small intestine and a low concentration in the blood. Question 17. Why don't root hair cells contain chloroplasts? Why don't root hair cells contain chloroplasts? What's a chloroplast used for, and where is that root hair cell? And I don't mean hair on your head, I mean a root hair cell in a plant. And the reason is they are underground, so cannot absorb light for photosynthesis. Those root hair cells will not photosynthesize. It's this palisade cells inside the leaf, the palisade cells. Okay, moving on to question 18. Which differentiate first, plant cells or animal cells? Which differentiate first, plant cells or animal cells? Okay, let's have a look at our answer. 50-50 on this one. It is animal cells. Plant cells can differentiate at any point in their lifetime. Animal cells differentiate very early on. Question 19. What is a countercurrent system found in fish? So the water flows over the fish gills in a countercurrent system. What does countercurrent system really mean? And we're looking for our answer now. Blood flows in the opposite direction of the water flow. It creates an effective diffusion gradient. So if the blood flows this way, the water flows this way. Creates a diffusion gradient at every point. Okay, moving on to question 20. How does oxygen exit the leaf? So movement of molecule questions. How does oxygen exit the leaf? Hopefully you're shouting at the screen, and the answer is diffusion through the stomata. Now, I will allow 2 points here, 1 for diffusion and 1 for stomata. Moving on, question 21. What are stem cells in plants called? What are stem cells in plants called? We think about Christmas. What do we say at Christmas? What are stem cells in plants called? And the answer is, of course, meristems. Just like at Christmas when we say Merry Christmas. They are meristems. Question 22. What is the movement of particles from a low concentration to a high concentration called? What is the movement of particles from a low concentration to a high concentration called? Okay, low to high, it's active transport. Question 23. What is the cell wall made of? What is the cell wall made of? And the answer is cellulose. Well done for those people who got that one. Moving on to question 24. What happens in the cytoplasm? Okay, we're talking about our basic Year 7 understanding of what happens in the cytoplasm. You might want to extend your answer with a bit of Year 9 and 10 knowledge. What happens in the cytoplasm? And the answer is chemical reactions. And we know now that those chemical reactions actually happen in the mitochondria, the ribosomes and the chloroplasts, and they're all floating around in the cytoplasm. So our basic answer is chemical reactions, and those chemical reactions actually take place in those three subcellular parts. Okay, on to our 25th question now. Organelles, that's the word I was looking for, subcellular organelles. 25. What makes leaves green? What makes leaves green? Okay, and we're looking for the answer of chlorophyll. And when you've done the later topic of B6, you know that you can get chlorosis of a leaf when there is not enough magnesium and it goes yellow, okay? But it's the chlorophyll that makes the leaf green. We're now halfway through our quiz today, our B1 quickfire question quiz. And if you would like to take a short break right now, maybe have a chat, catch up, have some snacks, and then come back to the second part. Okay, let's move on to question 26. There are two benefits of an electron microscope. Okay, they're very expensive. We couldn't possibly afford to have one in school, thousands and thousands of pounds. But what are two benefits? Really tricky on this one. We must be specific to what the course content wants us to include. What are the benefits of an electron microscope? And our answers are 1 point, higher resolution. It means we get a really sharp image, a really clear picture. And higher magnification. We can see more of those subcellular structures. I said subcellular organelles, that's also okay to say, and I did lose my memory for a short time. So high resolution, 1 point. High magnification, 1 point. Moving on to question 27. What do bacteria have instead of a nucleus? What do bacteria have instead of a nucleus? No nucleus in a bacteria. What do they have instead? This is a 2-point question. Got to get our technical language. Okay, we're looking at for answers of a single loop of DNA for 1 point and rings of plasmid for a 2nd point. A loop of genetic information also gives you a point. Question 28. How does glucose move into the blood after reaching an equilibrium with the villi? Now earlier we said that the glucose starts off moving by diffusion into the blood, and now we're saying how does glucose move into the blood after reaching an equilibrium with the villi? Okay, so it's reached equilibrium. That means there's the same concentration both in the villi of the small intestine and in the blood. So to get that last push across, active transport. Question 29. In the osmosis investigation, that's the potato one, why do some potatoes get heavier? What happens to those potatoes? In the osmosis investigation, why do some potatoes get heavier? Could be a 2-point question. Okay, and the answer is that the concentration of water in the solution was higher than in the potato, 1 point. So the water moved in and it got heavier, 2nd point. Okay, the concentration of water was higher in the solution than it was in the potato. So the water moved in and it got heavier. Let's move on to question 30. Hopefully you're still with me and enjoying this quiz time. If the If the eyepiece lens has a magnification of times 10 and the objective lens is times 8, we're in our microscope now, what is the total magnification? So we've got the eyepiece lens at the top of the microscope and that is times 10 magnification. The objective lens near the stage is times 8. What is the total magnification? And the answer is times 80. We multiply those two numbers together. Okay, we don't add them, we multiply them. Moving on to question 31. How are the villi adapted for diffusion? Now we've got 5 possible points here. If you can remember your points for alveoli, they'll all work, but we need 1 extra point for our 5th point. Okay, let's go through our marking. We've got 1 mark for large surface area, 1 point for large network of blood capillaries, 1 point for that moisture that allows the gases to diffuse, 1 point for thin, they're only 1 cell thick, and 1 point for those microvilli. Now microvilli increase the absorption rate by about 200 times, so even though they're micro, they're incredibly powerful. Possible 5 points there. Question 32. How many pairs of chromosomes are there in human cells? How many pairs of chromosomes are there in human cells? Hopefully you're shouting the answer at the screen. And our answer is 23 pairs. We always learn about humans, but we could have any question in the exam and we could have any number of chromosomes because different organisms have different numbers of chromosomes. Question 33. What will happen to a potato put in a highly concentrated sugar solution? So I have a potato, I've got a very concentrated sugar solution, very high in sugar. What's going to happen to that potato? Okay, and the answer is it will lose water and get lighter. Notice I didn't write loose water. Okay, loose has 1 O. Commonly people put 2 O's, which is loose. Okay, so it will lose water and it will get lighter. The water will drain out of the potato by the process of osmosis. Question 34. What is a drawback of using bone marrow stem cells? What is a drawback, a negative point, a disadvantage of using bone marrow stem cells? There is more than 1 possible point here. And the answer is limited uses. We can only get a certain small number of different types of cells from it, painful removal, and possible infection. 3 possible points there. Question 35. What is the 3rd stage of the cell cycle? This is the final stage of the cell cycle. What is the 3rd stage of the cell cycle? We've had mitosis, we've had interphase. What is the third stage? And of course it is cytokinesis. Cytokinesis. Question 36. What is a benefit of embryonic stem cells? Again, more than 1 possible answer here. What is a benefit of embryonic stem cells? Okay, and possible answers can make any kind of cell. Oh, the other option which isn't on there is won't be rejected. Okay, won't be rejected. And moving on to question 37. What 2 cells are used in therapeutic cloning of an embryo? This is a tricky process, a bit like adult cloning where we looked at Dolly the sheep, and everybody forgets this process. What 2 cells are used in therapeutic cloning of an embryo? It's known as an ethical embryo, but what cells do we use? And for your 2 points, you've got a skin cell and an empty egg cell. Skin cell has the pairs of chromosomes, it's quite a simple cell, hasn't differentiated too much, and the empty egg cell with the nucleus removed is what the skin cell is then put into. So 2 points there. Question 38. Which movement of molecules must be through a partially permeable membrane? Okay, so it's partly permeable. Some things can go through, some things can't. Sometimes they call it a semi-permeable membrane for the same reason. Some things can go through and some things can't. And which movement of molecules must go through that membrane? 3 to choose from. And the answer is osmosis. Well done for that one. Okay, moving on to question 39. What happens during interphase? So we're in the cell cycle. What happens during interphase? It's that first stage of the cell cycle. What happens in interphase? And the answer is the chromosomes, or the DNA, copies, doubles, replicates or duplicates. You don't need to go for all of those words. One will do. Ready, just like that photocopier, to then split the cells into 2 identical cells. Question 40. In the osmosis investigation, the one with the potatoes, why do we convert the change in mass to a percentage? Why do we convert the change in mass to a percentage? And the answer is, so we can compare all the results. If the potatoes started with very different masses and we just looked at how much mass was gained or lost, they are not comparable results. By looking at a percentage change in mass, we are able to compare all our results nice and fairly. Question 41. Where does the energy for active transport come from? Where does the energy for active transport come from? Remember, energy can't be created or destroyed. So where does that energy come from? Possible 2-point question, I think. Possible 2 points. Okay, let's have a look. It's respiration in the mitochondria. Okay, so 1 point for respiration and 1 for mitochondria. Question 42. How could we use meristem cells? Really tricky one this time on the B1 quickfire question quiz. How could we use meristem cells? These are the ones from plants. 2-pointer. Okay, we could clone rare species of plant. If those plants are in danger of becoming extinct, we could use meristem cells to clone them and increase those population numbers. We could also use them to grow crops with special features. Maybe there's medicinal purposes for them, and we need lots of them to get that medicine to the people. We could use meristem cells. Question 43. If you're not enjoying this, we're nearly at the end. Well done for sticking with me. What is the purpose of the cell cycle and mitosis? So mitosis is the second stage of the cell cycle. People tend to forget the phrase cell cycle, so I've reminded you that mitosis is the major part of that. So what is the purpose of the cell cycle and mitosis? And the answer is growth and repair. You might have put to make identical cells. That's also worth a point. Growth and repair. Unlike meiosis, which is used to make gametes. And question 44. If active transport is an active process, how would you describe diffusion? If active transport is an active process, meaning that it actively uses energy, how would you describe diffusion? There's a type of immunity with this word at the beginning, and no, it's not herd immunity. And the answer is, it is a passive process. That means it doesn't require energy, it just naturally happens. Question 45. In the osmosis investigation, the one with the potatoes, why do we roll the potato on a paper towel before finding the mass? We don't crunch it down on the paper towel, but we just gently roll it over. Why do we do that? And the answer is to remove excess water that could affect the mass reading. Okay, all that extra water dripping about going onto the scales would increase the reading on the scales. So we remove the excess to get an accurate mass reading. Question 46. What kind of cells are produced in the cell cycle and mitosis? I believe I've given you the answer to this in a previous question. What kind of cells are produced in the cell cycle and mitosis? They are cells for growth and repair, but how would we describe those cells? And the answer is identical cells. They are diploid. They have pairs of chromosomes. Possible 2 points there because they're identical and they're also diploid, and we think of that as meaning double with the dip at the beginning there, which means pairs of chromosomes. Question 47. What process do mineral ions use to move from the soil into the root hair cells? Now, it would be different if you had suddenly added lots of fertiliser to the ground, but we're thinking of soil in its natural state. What process do mineral ions use to move from the soil into the root hair cells? Again, not hair cells in your head. No, no, no. Their root hair cells are in the plant. And the answer is active transport. Of course it is, because they're in such low concentration in the soil there, and we call that the greedy one. It's trying to get every last mineral ion into that plant. Question 48. How can you change 61,000 into standard form? How can you change 61,000 into standard form? Comes up in the microscope questions, hence in this topic, if you're wondering how this relates. Give you a little bit of time, a bit of a tricky one. Might need to write it down. And the answer is 6.1 times 10 to the 4. You can see there I've moved the decimal place 1, 2, 3, 4 times. 6.1 times 10 to the 4. 61 times 10 to the 3 is not possible. Standard form is always one number, a decimal point, any further numbers, and then times 10 to the power of how many times you've moved that decimal place to the left. The only answer is 6.1 times 10 to the 4. And question 49, the penultimate question. Oh my goodness, we're probably so sad that this quiz is finally coming to an end, but don't worry, next week we're looking at B2. What is the movement through the phloem called? Early on we had a question about the xylem. We learnt that the xylem had the flow of minerals and ions, so the phloem is obviously something else. And what's that movement called? We learnt that the xylem, it was transpiration. What could it be called in the phloem? And the answer is translocation. Transporting those sugars made in photosynthesis to all the different locations around the plant that need it for respiration. Excellent, good one. And the final question of the B1 quickfire question quiz is what happens during cytokinesis? What happens during cytokinesis? And the answer is The cell membrane splits, forming two separate cells. Cell membrane split, forming two separate cells. The whole point of the cell cycle is to copy that identical cell, and cytokinesis is that final split of the cell membranes to form two separate identical cells. Tally up your scores if you want to. Send me a message on Show My Homework and let me know how you did compared to your friends. I would love to hear that information. I'd love to know if you enjoyed it or if I'm wasting my time. You can always show your appreciation by hitting subscribe. If you don't like the Quizmaster voice, it's probably explains the reason why I don't allow comments to be written on my videos, okay? So you can keep those ideas to yourself and we'll laugh about it when we're back at school. But remember, this was the B1 Quickfire Question Quiz. Have a great day. Hope you enjoyed it. Thank you.